Okay, well this is the model that I was talking about. And it shows the double helix coming around, and in some places you have a wide gap, and in others you have a narrow, just like in the real molecule. You have your phosphate groups on the outside, along with the sugars, and then you have the different colored on here, of course the real thing's not colored, um, cytosine and guanine, or adenine and thymine. And that's a pretty good model of the way DNA looks when it's not all crushed together inside of a chromosome. Okay, so tonight I'm going to talk about DNA analysis, how we go about using the information from the last time in order to see what happens. So DNA analysis is often referred to as DNA profiling or DNA fingerprinting. Both of those terms are used interchangeably. It's used in a wide variety of areas. In forensics, you're matching samples to a suspect. In the trial of O.J. Simpson, they showed the results. They showed that a blood smear that was found at the scene of the murder was the same, had the same DNA basic structure as O.J. Simpson's blood had. So that was one clue. And it's also used in forensics to prove that it was not somebody who did something. There have been a number of cases now that have been overturned. People have served many, many years in prison, and then they found out the DNA proved they were innocent. So it's become extremely important. I think it was in 1963 that it was first figured out. Didn't come into a lot of use until the late 70s. And in the late 70s, they also figured out not only can we use this to make pieces of DNA, we can use it to recombine the DNA using an enzyme called ligase. Remember, ending in ASE means it's probably an enzyme. And change the DNA for organisms. Do realize that this was used, back in the 70s up until fairly recently. And now in forensics, it's become so easy to find out the entire DNA, to sequence an entire strand of DNA to know a person's complete structure of the DNA, that is now used because it's less expensive, it's faster, uses less material, and is less equipment in order to do that. Still used a lot in laboratories and in research. It's been used in genealogy, finding out who's closely related to whom. And there's online companies that will trace your DNA to see if you are related to people in certain parts of the world. And it's been used in paternity testing to find out who the real father is, as they like to show on some of the TV shows. It is used a lot in medical research, used in immigration eligibility, determining whether a person is eligible to immigrate. It's used in agriculture an awful lot, trying to improve crops and crop yields. Used in botany and zoology for research. Used in food analysis. They found out, for instance, that some fast food restaurants, their hamburgers were not entirely beef. And sometimes they were putting in other animals, and sometimes they were putting in soybeans or other fillers just to increase the volume and decrease the cost of making them. The first step that's used in DNA analysis is to find a sample to analyze. Now, if it's a person, most often they take what is called a buccal swab, and that's where they take a Q-tip and put that on the inside of your cheek. Does not require hard scraping. I always tell my students, you don't have to scrape hard. I don't want to see the Q-tip coming out the other side of your skin. But you just do that and you'll get some cells. Other possibilities include samples from blood, saliva, semen, vaginal lubrication, fluids or tissues from personal use items such as a toothbrush or a razor blade, wherever you can get a sample. And they can use stored samples such as a banked sperm or biopsy tissues that have been taken from surgeries. Once you have a sample, then you've got to isolate the DNA. In order to do that, you've got to break down the cell membrane and then also the nuclear membrane so that the DNA can come out. Then you dissolve it in some solvent, and then you use some sort of extraction procedures. There are organic, Helix, solid phase, differential, different kinds of extraction procedures to get a sample of DNA. Realize that when we use blood, most of the blood is red blood cells. But human red blood cells do not have DNA in them. So you end up discarding those. So what DNA do we get from blood? Mostly it's the DNA that's in white blood cells. No, entirely. Which is a very small constituent within the blood. When you spin down blood, you'll have the top part is serum, the bottom part is red blood cells, and then in the middle there's a very small area that will be your white blood cells. The next step is called RFLIP analysis. RFLIP, or RFLP, that's an acronym for Restriction Fragment Length Polymorphism. Different lengths of the DNA when it gets chopped up. My wife was a medical professor of medical genetics. She told me nobody calls them RFLIPs. However, I keep running into it everywhere. So maybe some people must be calling them RFLIPs. In this procedure, they take restriction enzymes and use them to cut the DNA at specific points. Different enzymes cut at very, very specific different points. Then they use what's called gel electrophoresis, which we're going to get into later, to separate the different size pieces of DNA. Then, if they wish, they can do further analysis with something called polymerase chain reaction, or PCR, which is a way of increasing the amount of DNA so that you go through a procedure that involves frequent cooling and heating and different enzymes working to increase the number of copies you have of the DNA. When I've done these myself, we would let that run overnight, and it would end up giving about 1 million times as much as DNA as what you started with. One potential problem for PCR is you have to make sure you got the right DNA, because if you have a sample that's contaminated and you happen to get DNA from a different cell, then you end up with a lot of copies, but they're not the ones you're supposed to be looking at. And that's one thing they have to be very careful of when using that. Then they can follow that PCR that increases the amount of DNA you have with things such as the southern blot, which is a way of spreading out the DNA based on size, similar to the gel analysis that we'll be doing. As I said before, there's a variety of restriction enzymes that are available. These come from bacteria. Bacteria have these restriction enzymes because they will chop up the DNA of a virus and keep the bacteria from being killed or taken over by a virus. We know of more than 100 different restriction enzymes. I've given you a short list here of some of the more commonly used ones. Their names, the third one is EcoR1. That may sound a bit weird, but that name comes from the name of the bacteria that it came from. EcoR1 came from, geez, my mind just went blank. Bacteria. I'll come back to it. Escherichia coli is the bacteria that have it. So, and then in the third column, it gives you the specific sites on the DNA that it looks for, and then the average size of the pieces that are made by it. And of course, that size of the pieces is going to vary with the DNA that it's analyzing. Last time we talked, I said that adenine always bonds to thymine, so it's A to T, T to A. Guanine always binds to cytosine, always G to C, C to G. They're held together by hydrogen bonds from the two different strands of the double helix. And I showed you this before, and you can see there the ATGC combinations throughout. And it's the number of these combinations that will vary a lot from one sample to another. So for example, EcoR1 from Escherichia coli, it's a restriction enzyme that cuts at this specific site, and only this specific site. From the 5' end, it will cut between the G and the A if you have the complete sequence GAATTC. If it doesn't have that sequence, it's not going to cut it. And of course, the complementary strand, the other part of the double strand, is going to have the reverse of that. And so it's CTTAAG, but if you read that backwards from the 5' end, it's GAATTC. So when those come apart, when the restriction enzyme is like a molecular scissors, and it goes in there and cuts them apart. And so when you do that, you'll see that you're going to have ends sticking out. And those are called sticky ends. And the first one here is showing the result for the sticky ends with an EcoR1. Now there's another restriction enzyme, SMA1. When that digests, it looks for the code CCCGGG. And it's going to cut between the C's and the G's. And so you end up with what are called blunt ends. The sticky end, if you're doing an experiment where you need to put the DNA back together again, sticky ends will go back together much better than blunt ends will because of the overlap. So if you get this piece of DNA and then you add ECO1 to it to digest it, how many fragments are going to be formed? Look through there and see if you can tell. How many places will it cut that double-stranded DNA? And up here, I've given you again, it's GAA-TTC from the 5' M, which is the top strand there. This is the audience participation part.
2 places, right?
Couldn't quite hear that.
2 places?
Okay. How about the second one? Wait a minute, I didn't give you the second question. How many would be made, how many fragments by an SMA1 digestion?
Two again?
Say again, please.
2?
2? Let's find out. So the blue indicates the sites where ECO1 would digest. And there are 2 sites that for the blue that would be broken apart. With 2 sites broken, that means you're going to have 3 separate pieces of different lengths. For the SMA1 digestion, which is shown in yellow, there's only 1 site where it'll digest. And so that will break it into 2 segments of different lengths. Any questions so far on that? Okay, we'll move on. How do you then analyze that when you get these different fragments? I'm going to show you a presentation that I borrowed from mayfieldschools.org because it's a nice one. And it's got nice graphics. I don't have to go through making them all. And this will show DNA fingerprinting. There are many uses of restriction enzymes. With the restriction enzyme, we can cut the DNA like a pair of scissors. We can cut up the DNA from different people or different organisms and compare it. Why would we want to do that? As I said before, it's used in forensics. Oops. Medical diagnostics. Of course, now that we can sequence an entire DNA, it becomes even better. So you can actually develop drugs, we will, that are specifically made for a specific person, working just for them. Paternity testing, evolutionary relationships, how related are 2 organisms according to the theory of evolution. So how do we compare those cut up DNA fragments, and how do we separate them? You can't do it with a microscope. They're too small. We separate the fragments by size, and then we run it through a gelatin. Gelatin is usually, not always, but usually agarose. Agarose is made from algae, basically seaweed. And this shows a gel with different samples made by gel electrophoresis, separated by gel electrophoresis. So gel electrophoresis is a method of separating DNA in a gelatin-like material using an electric field. There is a charge on DNA, and so an electric field is going to cause it to move through the gelatin if it's set up properly. DNA is negatively charged. So when it's in electrical field, it's going to move towards the positive side. Opposites attract. And in the bottom diagram, we see that they put the DNA at the negative pole and it progresses across to the positive pole. And the bigger ones, which are heavier, more dense, are not going to go as far due to the charge. They don't move through the gelatin as easily. And so you end up separating by size, with the smallest ones going the furthest. So how does that help you in comparing the DNA fragments? The size of the DNA fragment affects how far it travels. As I said, small pieces travel further, large pieces travel slower, and they lag behind. Which means that by varying voltage, you can change how fast this works. And again, we see that the bigger pieces don't move as far. So this diagram shows you how this is done. In this case, they're using a vertical setup in the diagram here. Most of the gels I've run, we used horizontal gels. The top of the gel, right there, you can see there are welds in the gel that you put your samples in. And then when you attach electrical connections, then the sample allows the DNA to move through the gel. And I slipped for a moment there and said protein. This is also used with proteins. And then you have your final gel. Now, the gel has to be stained in order for you to see the DNA. And again, it's showing that the longer fragments are going to have more weight to them, so they're not going to go as far. And that's referred to as running a gel. Your different samples will end up having lines in different places. Where they're in the same place as another line, then that indicates that they're possibly the same DNA. So this is what the chart ends up looking like. And you'll run some known sample to get the sizes. Often, ethidium bromide is used to stain DNA. However, that's somewhat controversial. I have read that it is believed ethidium bromide can cause cancer. So in the schools, we quit using that. The Carolina Scientific Company makes their own special dye. It's called Carolina Blue, and that is non-toxic, and that's what we use. And the ethidium bromide fluoresces under UV light. One of the nice things about the Carolina Blue is you do not have to have a UV light in order to see it. And there you see the gel after when it's fluorescing under the UV light. And you can see which samples are similar and which are not. And it's all based on these restriction fragment length polymorphisms. Now we refer to them as alleles, but in actuality we don't know that that fragment codes for a specific gene. We just know that it is a fragment. Keep hitting the wrong button. So changes in the DNA sequence will affect where that cut occurs and gives you different size pieces. You get a different band pattern. And there are people who spend a lot of time looking at those gels, matching them up. And when my wife was doing her fellowship as a medical geneticist, she once spent a whole day in the room looking at those, not thinking about the fact that she's using UV light, and she came out with a very bad sunburn. So, it can be used, as we said, in forensics, comparing DNA sample from a crime scene with different suspects and victims. So, in this gel, we have on the far right a sample from the crime scene. And V are some standards that are run. And then we have our 3 suspects. So, do any of the suspects match, in your opinion, the crime scene sample? And if so, which one? Who would you arrest? Pardon?
Is anyone awake?
Oh, that's an excellent question.
I'm just thinking here, sir.
And unfortunately, something's wrong with, I think my earplugs fell out, and now I'm not hearing people.
Okay. I'll give it to you, Dave. Can you hear me?
Yeah.
Okay. So far, no one's
Maybe
heard.
suspect 2.
Suspect number 2.
Yay!
You
You
win.
are correct, because you see that
Heck yeah.
these 3 line up, but here, it's just these 2. That one band there and one band there, perfect lining up. And then these 3 line up perfectly on suspect number 2. So yeah, that's a pretty good case for feeling that suspect number 2 was the one who left that sample. Now, it doesn't say necessarily when the sample was left. I just watched a TV show, Bull, where the suspect had blood on his shoelaces that matched the victim. And his claim was that the victim's blood got there because she had a nosebleed the day before. And in the end, they got a confession from the person who really did do the killing. It wasn't the one they thought. Comparing blood samples on defendant's clothing to determine if it belongs to a victim. So here we have another actual one, the victim's blood here, the blood that came off of the defendant's clothing in those 3, and then the defendant's actual blood. And is there a match-up?
Oh, yeah.
I'd say yes.
Now, it could be. Notice how this second band here is light compared to these others. Well, that may simply be the quantity of DNA that was used. So you do have to allow for that. And you have to look to see, does it actually line up, even though it's lighter? And on that one, there are ones that don't match up. What's referred to as DNA fingerprinting, comparing the DNA banding pattern between different individuals, trying to determine whether somebody was guilty. in looking for unique patterns. The first case where it was successfully used as DNA evidence was in 1987, a rape case where the defendant was Tommy Lee Andrews, and they've got the samples here. So you have the semen from the victim, the suspect's, let me pull that out of the way, the suspect's blood, looking to see if it matches up, and then the victim rapist semen and the suspect's blood. And so it was decided that, yes, these matched up perfectly. So the female who had the rapist semen inside of her, they were able to prove that it was from Tommy Lee Andrews. And so he was convicted. Come on. There we go. This is from another murder trial. This one here is just standards that are run for comparison, as is the one at the very bottom. So we have samples of blood from the crime scene. That's the top three samples. So it's pointing out the similarities and the differences. And then a blood sample from the victim. And a second blood sample from the victim. And then the blood sample from the suspect. So does the suspect's blood sample match any of the blood samples from the crime scene?
Yeah, it looks like
Would you
Match.
say that suspect is guilty?
He was definitely there.
These do require some interpretation, and you can see why it ends up that we have experts to deal with this. But yes, we have some similarities. Both sample 1 and sample 3 match pretty closely with the suspect. And in this case, it was O.J. Simpson. Now, were you guys even alive during the Simpson trial? You don't know. I don't think you were. It was a while back. Does anyone know the result? No, before your time. O.J. Simpson, who was a major athlete and who was a star in many movies, and he was acquitted of murdering his ex-wife and her lover. And the main reason he was acquitted, main reasons, was the work of the police force. They did things to corrupt the crime scene. And so it ended up being thrown out. But then a civil suit was filed against O.J., and in that, he lost. And so he ended up having to pay. And then later, got arrested for some other crime and ended up going to jail. It's too bad. He was a funny actor. Anne Brown was the lady. R. Goldman was the man, if I remember right. Excuse me. But a very famous trial where the DNA results were actually ignored. In medical diagnosis, comparing normal alleles to diseased alleles. Sometimes the DNA causes a problem. So you have the alleles from two different samples. This is a test for Huntington's disease. Huntington's chorea is a very nasty disease. Used in paternity. So you have a mom, you have a suspected father, in this case two different suspected fathers, and you have a child's DNA. Is the child's DNA going to be the same as the mom's?
Someone take a guess.
No.
Hey, 50-50 chance. Sure, it's not going to be. Because the child got half of its genes from the mom and half from the father. Now, there's an exception to that. If you look at the mitochondria, remember that's the powerhouse in the cell. The powerhouse in the cell, the mitochondria, takes the glucose and turns it into energy for the cell. Well, the mitochondria are inherited directly from the mom. Dad's DNA won't be in there. So if you look at just the mitochondrial DNA, it would be the same between mother and child. But there's usually not a lot of concern about whether the mother is the real mother of the child. If she went through childbirth, then there's a good chance that's her child. But who's the real father? And that can matter a lot for money. So, the mother should not have the same DNA as the father. The child should have some DNA, same as the father. So when you look at these samples, you're looking to see, for example, this right here does not appear in either of the suspected fathers, but it does in the mother. So that's probably a section of DNA that came from the mother. So then you look at the others to see, is there any DNA in the child that was definitely not from the mother? Who's the father, F1 or F2?
F2.
Number 2.
Was that F2?
Yep, F2.
Okay. Yes, right here, you see, this piece of DNA in the child is not found in the mother's DNA, but it is found in F2. So yes, F2 is the father. And there's other places where they line up too. Of course, the child doesn't have all the DNA from the father, so there are places that would not be the same. Testing for evolutionary relationship that compared DNA samples from different organisms to measure their evolutionary relationships, how closely they might believe that they are related. It used to be all evolutionary theory was based on structures that they would find. When they started getting DNA evidence, the DNA is much more indicative. And so they're determining how closely related two organisms should be by how much similar DNA they have. And this is called cladistics, where they determine how closely related the organisms are. And they end up coming with family trees, essentially. This is something that is highly controversial. As a friend of mine said, all cars have wheels, but that doesn't mean they were made by the same person or that they're closely related. It's just a good idea. Wheels work well. So why is it that each person's DNA pattern is different from another's? And partly it's due to sections of what are called junk DNA, which doesn't code for proteins. I've always felt a bit iffy on this because the more we study, the more we figure out. And so sometimes we found out what we thought was junk DNA was actually doing things.
Yep.
The junk DNA is often made up of repeated patterns, like CAT or GCC. And each person may have different numbers of those repeats. You have 23 chromosomes, unless you have an abnormality, and many of the sites on those 23 chromosomes will have different repeat patterns. And so there it's showing one example compared to another, so that the CAT pattern is repeated more in the bottom one than the top one. Now, of course, the cut sites, again, depend on what enzyme is used, restriction enzyme. And so sometimes they may do several different restriction enzyme digests to get more to compare. And so here, the recognition gives you 3 different segment lengths. Oh. Okay, I don't know why it didn't show the second allele. But you can see how the different links come out at different places in the gel. Oh, now we're going to do the second one. More repeats is going to make longer piece of DNA. Heavier piece is not going to go as far through the gel. So the other 2 parts line up perfectly, but the yellow part came out differently because of the different number of repeating patterns. Okay. How are we doing? Okay. So what we're going to do tonight is go into the lab and make up an agarose gel. And the next Friday, we'll have a problem to figure out, and we will actually run the agarose gels to find out what happens. So now we'll go into the lab. Now, anytime you're going to run a lab, you should take a look at the instructions. Always. Make sure that you're not doing something incorrectly. It's very important. Now this is our Yeah, I'm not sure what they can see here. Okay, this is the power supply. You have your positive and negative ends. This is the supply to run two different chambers at once. This is the chamber where you actually have the gel. It has places where the electrodes come down inside so that there will be a charge. This is the form for making gel. And you actually make it in here. When it's being made, you add what's called a comb. Kind of looks like a comb. And it goes here to give wells for your sample to go into. Now, this is an older model of Carolina's electrophoresis apparatus, which means it has one particular problem that often messes up students. And that's that there's no end on here. It's open. But you're going to put gel in there, agarose, and you don't want it to come out. So you have to take masking tape and put it over the ends. Now, in the more modern equipment that Carolina is making, they have put barriers there so you don't have to worry about it leaking. And leaking is the major problem that students have when running these gels, or for that matter, anybody, because I also have had problems with leakage. You have to make sure that it's really well sealed. Okay. Oh, put the cone in. Now you only had to have the seal over the ends, but I like it everywhere because the comb tends to fall out. And this way the comb is held in place better. Okay. Now, for electricity to flow through this, you have to have a buffer in there. And the buffer that we're going to use is called tris borate EVPA. And so we have to make that up. Now, conveniently, this is tris borate EVPA, but it's a 20x solution. So we've got to dilute that to a 1x solution. So I'm going to make 250 milliliters of final solution. I want you to calculate and tell me how much of the tris we have to put in here that we can then dilute with water to make 250 mils of a 1x if we start with a 20x. Get those brains working. I can't tell if my earplugs have fallen out, so I'm not hearing anybody.
Yeah, well, I'm not hearing anybody yet either.
Okay.
Would you need
So you guys figure this out. We're just going to wait.
Hang on. Okay, go. What do you got, Rishi?
yeah, I was gonna say that you need 11.9 milliliters of the 1X, and then 238.1 milliliters of water. So you have a 1 to 20 ratio.
Of the 20X. Okay? Okay. I did want to mention you should always check to see any chemical you're going to use, what warnings it gives. So this one says, obtain special instructions before use. Do not handle until all safety precautions have been read and understood. Use personal protective equipment as required. Store locked up. Suitable for any general chemical storage. And storage code is green. General chemical storage in case of fire. In case of fire, use a dry chemical CO2 or appropriate foam. Dispose of in accordance with all federal, state, and local regulations, which anything you ever look at is going to make that last line. Know what the regulations are. But on the other side, it says, first aid. In case of contact with eyes, rinse immediately with plenty of water and seek medical advice. After contact with skin, wash immediately with plenty of water. If swallowed, do not induce vomiting. Seek medical advice immediately and show this container or label. In case of accident by inhalation, remove casualty to fresh air and keep at rest. And then it has a warning sign, and it says danger. May damage fertility or the unborn child. So this is stuff to be careful with. Now, as I've said before, any toxin, any poison, it's the dosage that matters. So this being 20X is much more dangerous than when we dilute it down to 1X. So I'm going to use a 10 milliliter pipette. I hope. Now that was 10 milliliters, but you said I needed 11.9, right?
That's correct.
So I see it better. So I'm just taking this up part way. So right now it's at 6.0. And so I'm just going to dispense there's one. Point 9. And immediately cap it because I don't want it sitting around. I'm sorry, you said 11.9 plus what?
Uh, 238.1 milliliters of water.
Okay. All right. Fortunately, in chemistry, we have something called QS. And Honestly, I don't know what QS letters mean, but what it means is bring it up to that volume. So I know I want 250. And this being a graduated cylinder, it accurately measures the markings on it. And there's 250. Now that substance is called parafilm, and it seals this quite well. And then I'll put my hand over it just to make sure, and invert this a number of times to make sure that it mixes really, really well. Alright, so the instructions then say that has to be stored out of sunlight. So I have a bottle I've already labeled, and it'll be stored in there when we're finished here. And we need to make up the agarose. And it says you can make 0.8 or 1%. So we're going to go with 1%. And that means you would put in 1 gram of agarose in 100 milliliters. And that's what we're going to do. We're going to make 100 milliliters of the agarose. So we need to measure out 1 gram. Using the balance, first making sure the balance is level, because this table is not. It's actually designed so if water were spilled, it would run towards the back instead of onto people. And get my weigh paper. This is a special paper, it's called glassine, so that things don't stick to it much. I like to fold it into quarters to make it easier to pour from it. So, we turn it on. We wait until it gives us a bunch of zeros. And then I can take this weight and put on it. And it should say 200, but it says 3.5270. Uh-oh. What does that mean? Go over here to unit and press that because it said ounces. And now it says grams and it says 100.00, which is what that weight has. So that's perfect. Well, And how much water did I say I needed? I mean, buffer? That's right, 100 milliliters. Okay, so that's our agarose, but it's not a gel, and it's not dissolved. So what we're going to do is microwave it. Interesting place to store those. And to microwave it, we have to keep a very, very close eye on it. Could you please turn out the lights? Because it will boil over. Right. Very, very hard to see it inside there. Can stop it for a minute. Stir, make sure it's mixing in. Yeah, it ran out of time. But that's what you have to do, is keep stirring it. And there is a light inside, it's just not real bright, and you're looking through this black weave. And it's boiling. And normally I would wear heat protective gloves like those, but since I have the plastic gloves on, they're doing a pretty good job. And I don't know if you saw that when I first took it out, but it was not bubbling, and the moment I shook it a little bit, it started bubbling a lot. So when you microwave a liquid, you need to be careful, because it will suddenly react when agitated. Now, it has to cool a little bit, and one of the things I never liked was they never say, how cool? Cool, but don't let it gel. Allow agarose to cool slightly, then pour your gels. after the gel sets, then you carefully pull out the comb and try not to break the gel any, because that comb is going to leave the wells that you put your samples in. How are we doing on time? It's time. It's time. You know, like I said, and this is a great idea. If they had told me at what temperature it's okay to pour. So I'm going to guess that it's okay, and we're going to find out if it's still going to go ahead and leak. It may do that. And you need to pour in enough that it's up near the top. And so far, that looks good. I don't see any leakage. I'm very happy about that. So later, after it's gelled, I'll pull out the comb, and then we'll store that in the fridge for use next time. Thank you all for watching.
Go.